Hello everyone, how are you all? Hope that you all are fine as well as healthy. We are learning biology, chapter number 5, and the title of chapter number 5 is Principles of Inheritance as well as Variation. Means principle of inheritance and variation. In last video lecture, we have talked about the introduction of this chapter number 5. In introduction, we said about the definition of inheritance as well as variation. Then you know that the definition of genetics. What is genetics? It is one type of science which deals with inheritance and variation. Okay, the father of genetics is Gregor Jonathan Mendel. If we talk about the father of experimental genetics, so you know the name father of experimental genetics will be T.H. Morgan, Thomas Hunt Morgan. Okay, Thomas Hunt Morgan did different type of experiments on fruit fly, and the scientific name of fruit fly is the Drosophila melanogaster. If we talk about the father of modern genetics, then father of modern genetics is the William Batson. Okay, what about inheritance? Inheritance is what? It is one type of process. Okay, it is one type of process in which characters will pass from parents to their progeny. Okay, this phenomenon or this process is known as inheritance. What about the variation? Okay, variation is nothing but it is one type of different character. You know, the all progeny are not similar to their parents. Okay, or all brother and sister are not similar to each other. It is one type of variation, means different type of characters. Today we are learning one extra lecture about the genetics. So in this lecture we will talk about some history of genetics, and in second part we will talk about the some scientists and their discovery. Okay, means the role of scientists according to genetics. This lecture is not in your textbook, means we are talking about the some extra information. Okay, we are not talking about your NCERT, because this lecture is not included in your NCERT. So in examination this lecture will not be asked. Okay, means the information or the questions will not be asked in examination from this lecture, but it is very important to know all the information connected with genetics. Okay, see the first paragraph. We can say that the history of genetics started with the work of Gregor Jonathan Mendel. His work on pea plants, underlined pea plant, published in 1866, described what came to be known as Mendelian inheritance. Okay, so underline this Mendelian inheritance, and remember the work done by Mendel is known as Mendelism. Right, the word Mendelism means what? Mendelism means the all different type of works done by Mendel in the field of genetics is known as Mendelism. In the centuries before and for several decades after Mendel's work, a wide variety of theories of heredity proliferated. Okay, in the year of 1900 marked the rediscovery of Mendel. Underline this, rediscovery of the work done by Mendel by different type of scientists like Hugo de Vries, Karl Correns and von Schermark. Okay, and in the year of by 1915 the basic principles of Mendelian genetics had been applied to a wide variety of organisms, most notably the fruit fly. Underline the name fruit fly, and the scientific name is Drosophila melanogaster. Okay, led by Thomas Hunt Morgan, T.H. Morgan, and you know the father of experimental genetics is T.H. Morgan, means Thomas Hunt Morgan, and his fellows Drosophilists. Geneticists developed the Mendelian model, which was widely accepted by 1925. So underline this, Mendelian model was widely accepted in the year of 1925. Alongside experimental work, mathematicians developed the statistic framework of population genetics. Bringing genetics explains into the study of evolution. So you know that genetics is 100% connected with the process of evolution, and there is another branch which is known as population genetics. Okay, now see the next one. With the basic pattern of genetic inheritance established, many biologists turned to investigations of the physical nature of gene So underline this, scientists wanted to know about the physical nature, means the structure, means the composition of gene. In the year of 1940s and early 1950s, experiments pointed to DNA, underline this word, DNA, as the portion of chromosome and perhaps other nuclear protein. See, DNA underline as what? As part, okay, as one type of part of chromosome, one type of portion of chromosome that held genes, means the DNA, which is one type of part of chromosome having different type of genes. A focus on new model organism like virus, bacteria, along with the discovery of the double helix structure of DNA in the year of 1953 marked the transition to the era of molecular genetics. Now see, another branch is there which is known as molecular genetics. Molecular genetics is one type of branch of molecular biology. Remember, molecular biology is different type of branch, okay, it is one type of branch of biology. So see, the one word is used which is discovery of double helix structure of DNA. You know what is the meaning of dsDNA? dsDNA means double strand DNA, okay. What is the meaning of ssDNA? ss means single strand DNA in the year of 1953. So you know that the model of DNA was given by Watson and Crick, means James Watson and Francis Crick in the year of 1953, marked the transition of the era of the molecular genetics. Now see, in the following years, chemists developed technique for sequencing both nucleic acids and protein. Nucleic acids is equal to DNA and RNA. You know that the DNA and RNA both are one type of nucleic acid. It is one type of acids which are found inside the nucleus are known as nucleic acid. But you know that the both are one type of genetic material. Both are one type of inheritance material. If we talk about the humans, in humans DNA and RNA both are present. But you know that only DNA is known as genetic material, while RNA having different type of other functions. We will learn about the types and functions of RNA in chapter number 6. So sequencing for both, okay, nucleic acid and proteins. Proteins are made up of amino acids, and the sequence of amino acid will decide the nature and properties, okay, in the characters of different type of proteins, while other worked out the relationship between the two form of biological molecule, the genetic code. So see, here there is a one word which is known as genetic code. Each and every gene having the special type of coding system which is known as genetic code. The regulation of gene expression. See, regulation, so gene will be regulated by something, okay, and the gene expression, if gene is there, yes, it is okay, but the expression of gene is also important, okay. So gene expression became a central issue in the 1960s. By the 1970s, the gene expression could be controlled and manipulated through genetic engineering. So see, there is another branch which is known as gene engineering or the genetic engineering. In the last decades of the 20th century, many biologists focused on large-scale genetic projects sequencing the entire genome. See, what is the meaning of genome? If any organism having total 25 genes, okay, so this all 25 gene is known as the genome. Genome is the subtotal of gene, means gene 1 plus gene 2 plus gene 3 plus gene 4, all gene, okay, all addition of all genes are known as the genome. So many biologists focused on the large scale genetics projects sequencing the entire genome. Okay, if for example, if human having 3 lakh genes, okay, then the sequencing, okay, sequencing of gene is very much important. If we are going to know about the sequencing of genome or sequencing of the gene, then we are able to produce the new organism, okay, with the help of various artificial methods. So this paragraph is also completed, and we can say that HGP, okay, human genome project, you will learn about HGP in the next chapter number 6. Okay, so see, the first part of this lecture is completed. In first part only 5 or 6 paragraphs are there and some histories are there, okay, and some important concepts or important words are also there. Okay, now see, if we talk about the historical developments in genetics, so total 3 different type of columns are there. Here, name of scientist and its contribution. Okay, I will only speak the important ones. Now see, In the year of 1485, Lindsay recommended the use of lenses for viewing the small objects. Okay, nowadays the microscopes are used. Okay, in the year of 1519, the scientists named Z. Hansen and H. Hansen produced the first operational microscope. Okay, again remember, the questions will not be asked, okay, in your examination of board. It is a one type of reference book. Okay, it is a one type of reference book for genetics, and this lecture is only for your information. Now see, in the year of 1665, Robert Hooke introduced the term cell and described the cork cell. So underline this, 1665, Robert Hooke, term cell and described the cork cells. Now see, 1668, F. Reddy disproved the theory of spontaneous generation of the maggots. It is one type of question of evolution you will learn in chapter number 7. If we talk about the 1672, Malpighi, classification of tissue, underline this, it is important. 1674, Anton von Leeuwenhoek improved lens system of microscope by the grinding process. Okay, now see the 1682, Crew described bladders and paws in wood and pith. 1694, J.R. Cabarrus conducted early experiments on pollination and reported the existence of sex in plants. 1694. Okay, in the year of 1700, Linnaeus classified the biological organism. Underline this, classification, Carolus Linnaeus. Okay, 1761, Colker hybridized various type of organism species of tobacco and conducted that each parent contribute equally to their characteristic of their progeny. 1779, Wolf, founder of embryology. Okay, now we will talk about the only important ones. 1824, Trochet showed that all plants and animals are composed of, means made up of, cells. Okay, now see, 1837, Robert Brown discovered the nucleus in the cells of flowering plants. It is very important. Okay, Robert Brown. Now see, what about the next one? We can say that in the year of 1838, Matthias Sleeden and Theodor Swan formulated the cell theory. The cell theory, okay, in plants and animals. Now what about the 1840, Perkinch gave the name or the term protoplasm. What is the meaning of protoplasm? Protoplasm and cytoplasm both are not same, both are the different. Cytoplasm is equal to, it is one type of dense liquid like structure which is located or situated between nucleus and the cell wall or the cell membrane. All different type of cell organelles are located in the cytoplasm. What about protoplasm? Protoplasm is equal to all living part of cell is known as protoplasm. Okay, what about the 1845, Eidon used photomicroscopy for the first time. What about the 1846, Nageli showed that plant cell arise from the division of the pre-existing cell. Underline this. Now see, 1858, Robert Vishro showed that animal cells arise from the division of pre-existing cell. What about the 1859, Charles Darwin put forth the theory of the natural selection. Underline this, Darwin. The Darwin is known as the father of evolution and gave the theory of natural selection. What about the 1862, Colliker used the term cytoplasm for the living material surrounding the nucleus. Okay, what about the 1865, Gregor Jonathan Mendel developed the fundamental principles of heredity, means inheritance. What about 1870, he's invented the microtome. It is one type of machine, okay, which will be used in the microtomy. In microtomy, different type of slide will be formed from the different type of tissue. What about the Mischer, 1871, isolated nucleic acid from the pus cells. Now, what about the 1875, Stansberg discovered cell division in plants and gave the term cytoplasm and nucleoplasm. What about the 1879, it is very important, Fleming introduced the term chromatin. So remember, the name chromatin was given by the scientist named Fleming. What about the 1881? E.G. Balbiani discovered the giant chromosome in the salivary gland of the Drosophila. Okay, giant chromosome means it is one type of extra chromosome. What about the Fleming coined the term mitosis? Okay. What about the 1883? Rouse proposed that chromosomes contain genes which are the units of heredity. Okay, if we talk about the next one, 1885, Simper introduced the term plastid. Then what about 1888? Theodore Boveri coined the term centrosomes. Okay, what about the 1888? Walders coined the chromosome. Remember, the chromosome name was given by Walder. Now what about the 1892? Hartwig proposed the protoplasm theory of inheritance. Okay, protoplasm is responsible for the inheritance. Next, what about the 1892? Theodore Boveri described the meiosis in Ascaris. Okay, Ascaris lumbricoides, it is one type of animal. What about the 1898? Camillo Golgi described the Golgi apertures, means Golgi body in the nerve cell of the barn owl. Okay, you know that the Camillo Golgi used silver nitrate. Okay, and he had observed the Golgi body in the nerve cell of barn owls. What about the 1898? Benda discovered the mitochondria in the spermatozoa and other cells. What about 1899? Altman term nucleic acid. 1900, Karl Korens, Hugo de Vries and Van Schermark rediscovered Mendel's law of inheritance. You will learn this in next video lecture. What about the Sternberg? 1901 introduced the term plasmodesmata. Okay, you know the plasmodesmata is also there. Now what about the 1902? Hugo de Vries, the term mutation. See, mutation is very important concept and the name mutation was given by Hugo de Vries. 1902, Walter Schotter and Theodore Boveri proposed that the chromosome theory of heredity and identified chromosome as carrier of genetic material. What about 1903? Walder proved centrosomes are the chromosomal region with which the spindle fibers become associated or connected during the process of mitosis. Okay, what about 1906? William Bateson. Remember, William Bateson coined the term genetics and proposed the concept of allele. So William Bateson is connected with the total three name. William Bateson is known as father of genetics. In the year of 1906, Bateson gave one word, genetics. So the genetics name was given by William Bateson and the concept of allele was given by William Bateson. And now William Bateson and R.C. Punnett, Reginald C. Punnett discovered genetic linkage in sweet pea. What about 1906? Johann Sein. Okay, so the term gene or genotype and phenotype. Gene, genotype and phenotype. These three words are very important words. Gene, genotype and phenotype. We will use these all three words in this chapter number 5. So underline gene, genotype and phenotype. These words were given by the Johann Sein in the year of 1906. Now what about the Karl Korens? 1909 reported cytoplasmic inheritance in Mirabilis jalapa. Okay, Mirabilis jalapa is one type of plant which is known as 4 o'clock plant. In Gujarati you can say that it is gulbas. What about the 1910? T.H. Morgan, Thomas Hunt Morgan, okay, studied crossing over and recombination Drosophila melanogaster and coined the term crossing over. What about the 1910? Okay, Nielsen Ehel proposed the multiple factor hypothesis. What about the Sturtevant? 1911? You will remember, see, Alfred Sturtevant is a student of Morgan. Constructed the first linkage map in Drosophila. So underline linkage map and remember this linkage map is connected with the HGP, means Human Genome Project. In Human Genome Project this linkage map is used. Okay, again T.H. Morgan 1915 co-related genetic studies with cytological studies. He put forth the theory of linkage and studied sex-linked inheritance in Drosophila. Allen 1917 sex determination in plants. 1921 parenting based isolated insulin. Okay, what about Bridges? So Genetic balance theory of sex determination. Crew reported complete reversal of sex in hands. H.J. Buller 1927 introduced mutation in Drosophila melanogaster by the X rays. And remainings are not important, but you will take a screenshot and you will write in your fair book. 1948 Porter described the endoplasmic reticulum. Robinson and Brown 1953 observed ribosome in plant cells. Okay, now see 1953. James D. Watson, Francis Crick and Wilkins proposed the double helix model of DNA molecule. Benzer, see 1955. Benzer described the fine structure of genes cistron, recon and mutant. Okay, so these three are the one type of definition: cistron, recon and mutant. These all are the concept of molecular biology. Okay, this cistron, recon and mucon you will not learn this in 11th 12th, but it is a concept of college. See, C.D. Dewey 1955 coined the term cytoplasm. 1955 Pellade observed ribosome in animal cells. Francis Crick 1958 explained the central dogma of molecular biology. See, central dogma is one type of very important theory. You will learn about it in chapter number 6. Okay. Now see 1950 Korenberg synthesized the DNA molecule in vitro. In vitro means outside the body. 1968 Woodcock and Fernandez isolated DNA from the chloroplast. Okay, next one. One 1993 Sharp Roberts proposed the split gene concept. Okay. What about the Smith 1993 studied the site detected mutagenesis. Now 1997 Wilmot cloned sheep. You know, Dolly is one type of cloned sheep. It is one type of sheep which was produced with the help of process cloning. So Wilmot 1997. Okay, so these all are the one type of concept. Okay, so this lecture is 100% completed here. Thank you very much.